The largest structure ever built by life on Earth is the skeleton of an animal 3 millimeters across. The animal builds in slow motion for 10,000 years, and the structure can be seen from orbit. The animal is a stony coral, order Scleractinia, class Anthozoa, phylum Cnidaria, the same phylum as jellyfish and sea anemones. An individual coral polyp is a small cylinder of tissue, 1 to 3 millimeters across, with a central mouth and a ring of tentacles that reach out at night to catch plankton drifting past. It lives inside a cup of its own calcium-carbonate, aragonite, the same mineral that forms pearls. Polyps bud clones of themselves. The clones remain connected. Over years and decades, a single founder polyp can produce a colony of millions, and the mineral cups fuse into the shared skeleton that we call coral. Stony corals first appear in the fossil record in the Middle Triassic, about 240 million years ago. The reef-building lineages alive today are the descendants. The polyp does not live alone in its cup. Inside its tissues, sometimes making up as much as 30% of its mass, are single-celled algae of the family Symbiodiniaceae, commonly called zooxanthellae. The algae photosynthesise. They pass most of the resulting sugar, perhaps 90% of it, to the coral. The coral, in return, supplies them with nitrogen, carbon dioxide, and a sunlit address in the warm shallows. The arrangement is ancient, well over 100 million years old, and it accounts for nearly all the color in a healthy reef. The coral's tissue is mostly transparent. The color is its algae. The arrangement is also finely tuned to water temperature. A stony coral in its home range has been living within a few degrees of its thermal limit for its entire evolutionary history. When the water gets too warm, the algae produce reactive oxygen species faster than the coral can clear them, and the coral expels the algae. Without them, the coral turns white bleached. Without sugar, it starves. The best-studied structure these polyps have built is the Great Barrier Reef. It stretches over 2,300 kilometers, 1,400 miles, along the northeastern coast of Australia. Its area covers 344,000 square kilometers, close to the size of Germany. More than 2,900 individual reefs and 900 islands comprise it. UNESCO listed it as a World Heritage Site in 1981. It is the largest single structure ever produced by living organisms, and astronauts on the International Space Station photograph it on almost every orbit. In 1999, Hoegh-Guldberg of the University of Queensland published a review paper in the journal Marine and Freshwater Research. It was titled Climate Change, Coral Bleaching, and the Future of the World's Coral Reefs. Using sea surface temperature projections from the CSIRO and the Max Planck Institute, the paper argued that recurrent mass bleaching would begin within 2 decades on the Great Barrier Reef. It also argued that most of the world's corals would not survive the 21st century in their present form. The prediction was specific: by 2030, bleaching every few years; by 2050, every year. The paper has drawn more than 10,000 citations since publication. Hoegh-Guldberg has since become the inaugural director of the Global Change Institute at Queensland. In 2017, Terry Hughes of James Cook University published a follow-up paper in Nature, with 50 co-authors. It analyzed 3 mass bleaching events on the Great Barrier Reef in 1998, 2002, and 2016, and found that the 2016 event had been the most severe on record, affecting 91% of individual reefs. The paper was the confirmation, 18 years later, of Hoegh-Guldberg's projection. The metric that now governs the science is the degree heating week, a measure of cumulative heat stress above a reef's long-term summer average. At 4-degree heating weeks, most corals on a reef will bleach. At 8, widespread mortality follows. Since Hoegh-Guldberg's paper, documented mass bleaching events on the Great Barrier Reef include 1998, 2002, 2006, 2016, 2017, 2020, and 2022. 4 of those events have landed in the last decade. The intervals between events have shortened from roughly 5 years to roughly 2. The cumulative footprint now covers nearly the entire reef. Not every bleached polyp dies. Warm-adapted symbionts can re-enter the tissue if the water returns to normal temperatures in time, and some coral species, notably porites and certain tabular acropora, are more resilient than others. The reef we are watching is selecting, in real time, for the polyps that can live at its new temperature. What is unknown is whether that selection can keep pace. The animal in the cup is the size of a grain of rice. The structure it has built can be seen from space. Both figures are true at the same time, and both are the same organism. Next, a smaller engineer still in the soil beneath our feet.